So our next speaker, which I'd like to introduce, is Dr. Lydia Siragusa from Molecular Discovery, and she's going to give us a talk about hopping around three-dimensional pocketomes to hide drug discovery processes. Please.
Okay, thank you, Giovanni. Thank you for giving to Molecular Discovery the opportunity to be here today. Okay. And good afternoon to everyone. I am Lydia from Molecular Discovery. And today I'm going to present an approach developed by Molecular Discovery that allow to browse collection of protein pockets, which we call pocketome, in order to give useful insight for drug design processes. The initial rationale was that over the years, we moved from analyzing the orthosteric site of a protein to analyzing the allosteric site. It should be.
Obviously, it's not.
Okay. Maybe more here. Yes. Okay. Okay, sorry. So the initial rationale I was saying, it was that over the years, we moved from analyzing the orthosteric site of the protein of interest to analyze the allosteric site, and then to analyze all the possible sites of a collection of protein. With this in mind, the BioGPS approach is able to detect pockets on a protein structure, to characterize them, and to compare them in order to give a cross-relationship map of protein pockets for several purposes, like ligand repurposing, ligand selectivity, or target triage. Okay. So by briefly summarizing the BioGPS architecture, we can see that it's composed by mainly 4 parts. The first one is an algorithm for pocket detection, pocket characterization by using molecular interaction field, comparison, and then analysis of results by using statistical analysis. Of course, BioGPS already possesses a pocketome of about 800,000 pockets with their pre-calculated molecular interaction field, pocketome that is ready for large-scale virtual screening. Each pocket in the pocketome is annotated with different data of a crystallographic nature, like the presence of a ligand inside, or a cofactor in the proximity of the pocket, the resolution, or biological data, like the pathway in which the pocket is involved, the presence of a cross-translational modification in the proximity of the pocket, or the disease in which the protein and so the pocket is involved. And lastly, the BioGPS provides a runaway server that allows the user to launch large-scale virtual screening on external CPU. Today, we will focus on the first 2 steps, and then we will see some application. The BioGPS algorithm starts with the pocket detection step, where the protein is plunged into a 1 angstrom grid. All the points are detected in concavities of the protein. For these points, the barrenness is computed, and they are clustered in groups. To this group, erosion and deletion are applied to return the pocket. Then the pocket is characterized by using molecular interaction fields that represent the non-bonding interaction that the cavity is able to have with a putative ligand. So hydrogen bonding acceptor and donor, and hydrophobic, and of course also the shape. The molecular interaction field are represented as color surface, as we can see here in the picture. And to simplify their complexity, from a computational point of view, of course, we select some representative points for each field. The number of the representative points is proportional to the energy of interaction. Then all possible combination of 4 points are generated. Each combination is called quadruplet. And then all possible quadruplets are stored in the common reference framework. That is the final representation of the cavity, where each bit is a quadruplet. Once we have the pocket characterized by using the common reference framework, this pocket can be compared with a ligand characterized in the same way, or with another pocket. BioGPS, in this case, search for similar quadruplet. When it finds two similar quadruplets, it overlaps them, returning the superposition of the pocket and the interaction of the pocket, and also a series of similarity score, one for each contribution, shape, acceptor, donor, and hydrophobic, and also a global similarity of the two pockets. This type of approach, based on the similarity of molecular interaction field, it is used especially when we are searching for unexpected similarities of pocket, so proteins that are not related in terms of sequence or folding. But BioGPS is also a faster way to compare pocket that is based on the residue features on the Blossom 62 matrix. Again, the two pockets are aligned in three-dimensional manner. And then we have a similarity score and also an RMSD. This second approach is particularly useful when we want to select some representative pockets, for example, in a molecular dynamic trajectory. Lastly, with BioGPS, it is possible for a collection of pockets to compute these descriptors that are energetic or geometric descriptors, and then to compare this pocket by using statistical methods such as principal component analysis. Shifting the attention on the BioGPS pocketome, first of all, I would like to highlight that we are using the biological assembly of protein data bank to build the pocketome and not the asymmetric unit, which is the smallest portion of the crystal structure. This is because sometimes the asymmetric unit is not complete, and so the pocket detection will result to be not correct, since the pocket is also not complete. Following these criteria, we have this pocketome of about 800,000 pockets, starting from 120,000 biological units belonging to 34,000 proteins. 111,000 pockets contain a ligand, for a total of 24,000 ligands in the database. This database can be consulted and pockets can be inspected and used for large-scale virtual screening. Also by creating a focused subset of pockets accordingly to the purpose of the study being carried out. And in this regard, here we can see some experiences where BioGPS was applied to build custom pocketomes. Just I will go very quickly. The first one is from 10 years ago, where the authors of this study used BioGPS to discriminate catalytic machinery that have the same catalytic triad, but they are able to catalyze different reactions because the environment was different. And so with the use of molecular interaction fields, they were able to discriminate the different enzymes and to build mutants that were used for industrial purposes. Then in 2016, BioGPS was successfully applied for finding new molecules in the low micromolar range via drug repurposing approach. And then during last year, we have built Chromatic, that is a coronavirus pocketome, where we have detected and characterized pocket on all the proteins of coronaviruses in order to understand the evolution of these pockets in this organism and to search for pan-coronaviruses inhibitor. We have also built a pocketome with autospot therapeutics for allosteric phosphosite involved in post-translational modification. And then in the following slide, I will present you the most recent work about E3 ligases, ALLODD, the GPCR's pocketome, done with Ijen and Sonia, and then the cancer PPA interactomes. Okay, so during last year, there have been many innovation in the protein degradation field, and the ability to guide and redirect the protein degradation by recruiting an E3 ligases and a protein of interest is a new therapeutic modality, which is arising much interest as an alternative to canonical small molecule or antibody molecule. For this reason, the E3 ligases are becoming a very interesting therapeutic targets. And despite there are several cases, and to exploit the potential of the three ligases, PROTAC technology has been developed. PROTAC are molecules with two moieties that are able to bring a protein of interest in proximity of any three ligases, and so to lead the protein degradation of this protein of interest. Despite there are several well-functioning examples of PROTAC, only 2% of available E3 ligases are used to make PROTAC, and in particular 10 or 13 E3 ligases that we can see here in the apple pie out of 600 E3 ligases are used to make PROTAC. So in order to try to broaden the view on the three ligases, we've built ALLODD, that is E3 ligase pocketome navigator, an accurate platform with E3 ligases pockets and molecular interaction field that can be used to select new potential E3 ligases for designing PROTAC and also new ligand ideas. To build this platform, we have followed this pipeline. Starting from Hubia web server, we have selected all the E3 ligases UniProt code, and then we have searched only for those one having a crystallographic structure in the protein data bank, and they are about 30% of the 600 available. Then on this crystallographic structure, we have detected pockets, characterized the molecular interaction fields, and performed a null against stall analysis where all the pockets were compared with the entire pocketome in order to reveal some cross-relationship among E3 ligases that can be used for drug repositioning of ligands. And in the last two steps, we have also assigned to each pocket a protectability score, and also we have screened this pocket against liganded pocket, so pocket having a ligand inside, and also to give idea for drug repurposing approaches. Focusing on the pocketome, here in the plot, we can see all the three ligases for which we have a crystallographic structure. They are colored accordingly to the subfamily they belong to. And of course, greater the section, greater the number of pockets that we have for that E3 ligases. And for each E3 ligases, here I report the example of Cerebron, that is the most common one. We give an identity card with an overview like the UniProt code, the PDB code, the number of pockets and the location of pockets that we have highlighted. And also for each pocket, some three-dimensional descriptors, the cross-relationship with other E3 ligases, and also some bidimensional information like the tissue expression and involvement in disease that we have extracted from Human Protein Atlas. Focusing on the three-dimensional descriptors, the first one is, of course, the ligand binding, because among this pocket, we have about 3,000 pockets. Some of them are already being studied for potential binders, not necessarily PROTAC. But this is, of course, an excellent starting point for the design of a PROTAC. In this case, we can see the subfamily that are most liganded, the MDM, that is widely studied subfamily, but it's very interesting also the BTB subfamily that have several ligands, but to date, none of its E3 ligases is used for make PROTAC. The second descriptor is the protein-protein interaction proximity, because of course, if one of these regions is close in a crystallographic structure to a potential protein of interest, this is a good starting point for the design of PROTAC also. And at these purposes, of course, we have excluded all the proteins that are part of the ubiquitination machinery, so all the pockets that are not in a potential protein of interest. And the third descriptor is the solvent exposure, because keeping in mind what we have seen before, the pocket, in order to be targeted by a PROTAC, should be sufficiently exposed to the solvent. And for this reason, we have computed the solvent exposure for regions already targeted by PROTAC in gray, And we have assigned a good score, a success score, to the green one that have the same range of solvent exposure. The sum of these three descriptors gave us the protectability score. And here, for each subfamily, we can see the distribution of the protectability score. And then we have focused our attention, especially on those three ligases having a score of 3 or 2. In this slide, I will briefly summarize the application of the ALLODD that we have done. The idea was that these two A3 ligases, TRIM24 and 33, in our cross-relationship maps, resulted to share a high percentage of molecular interaction fields. This means that as a site in the A3 ligases, the two sites are really similar. So the question was, maybe we can reuse the ligand of the 24 for targeting the 33. And this is what we have done in the pipeline here on the right. We have performed a cross-docking of ligand of 24, also optimizing this ligand for the ligand binding of 33. We have performed an in vitro assay at the University of Perugia by selecting two molecules that were able to bind the TRIM33. And then with the University of Siena, we have obtained the first crystal structure of TRIM33 with a ligand inside. It is the first E3 ligase structure with a new ligand inside. So this example show that this drug repurposing approach that we are applying in the E3 ligase cross relationship can reveal new opportunities for product design. Moving on the second example that Nxera Pharma is doing in collaboration with Molecular Discovery, they focused on GPCR allosteric structure. Indeed, they have highlighted 24 allosteric sites in GPCRs for a total of 62 non-covalent allosteric small molecules. The purpose of this analysis was to map these sites and to understand geometric and energetic descriptors of these sites, and also to build a cross-relationship map of allosteric sites in a drug repurposing perspective. So first of all, they have generated an ambiguous nomenclature of the sites, and they have applied three methods for detect pockets that we can see here in the plot. And we can see that BioGPS is a very good success rate in retrieving GPCR's cavity. In the second step, allosteric sites that were common in different GPCRs were compared in a drug repurposing point of view. Here we can see the example of cannabinoid receptor 1, the allosteric site, the blue one that is already targeted by a ligand. These allosteric sites were screened against all the other allosteric sites in GPCRs. And here we can see the top rank solution. We have in brackets two similarity scores. The first one is the three-dimensional similarity score, so the one obtained by BioGPS that represent the molecular interaction field similarity. And the second one is the sequence similarity. And it is very interesting to note how pockets sharing half of the residue, or even less, because I see also 36, can generate similar molecular interaction fields. Indeed, if we see the example of cannabinoid receptor and sphingosine-1 phosphate receptor, here we can see the superposition of the two pockets. They share 84% of molecular interaction field and only 57% of sequence. And in the other three pictures, we can see the superposition of the molecular interaction field as surface for the cannabinoid receptor and as mesh for the sphingosine-1. And in all the three interaction type, we can see some common region that can be used for the same ligand in a drug repositioning view. So in this example, we can see how focusing on molecular interaction fields and not only on sequence or folding can bring new insight into drug discovery processes. The last example that I'm going to present is an ongoing project in collaboration with the University of Bari. And here the question was, which are the changes that occur in a healthy cell when it goes in a tumor state? So we decided to focus on protein-protein interaction. Here I just simplified. In a healthy state, I can have 3 protein-protein interaction, and in tumor state, 1 is repressed and the other 2 are activated. We started from a study done in 2020 in ARGA group, where they have performed this pan-cancer mapping analysis of protein-protein interaction. By starting from cancer genome atlas, human protein atlas, and genome type tissue expression, they have built a bidimensional information of about 5,000 protein-protein interaction. The vast majority of them, about 4,600, are activated under tumor state, and only 200, about 200, are repressed under tumor state. And in this analysis, there are 12 type of cancer. This was our starting point. We have mapped this protein-protein interaction in the protein data bank in order to find complexes of this protein-protein interaction. And we ended up with 300 complexes. The vast majority, again, are activated interaction for the 12 cancer type. On these complexes, we have detected pockets by using BioGPS, of course, building the cancer-related pocketome of about 2,800 pockets. Here we can see the 12 type of cancer that we are analyzing. In the first column, the number of oncoprotein-protein interaction, we'll say, detected by ARGA group in 2020. In the second column, the number of complexes that we have found in protein data bank. And the last column, of course, the corresponding percentage. The question was, how can we prioritize this pocket for a drug design process? So this is an example of neuropilin that acts as a co-receptor for the vascular endothelial growth factor. This interaction is activated in kidney cancer and has an essential role in angiogenesis. So with BioGPS, we have detected pockets, and we can see 2 different routes. The first one, the yellow one, the pocket at the interface of the protein-protein interaction. By targeting this pocket, we can disrupt the protein-protein interaction, but also the other 2 pockets, the magenta one and the green one on neuropilin and vascular endothelial growth factor, can be used for PROTAC. Because even if they are already liganded, they can be used to design a PROTAC and to bring the protein to degradation, that we still have the final effect to disrupt this protein-protein interaction. However, it should be noted that in this case, also the yellow pocket can be targeted by a PROTAC, especially in this case, because for neuropilin, we already have some ligand in the yellow pocket. Following this criteria, this is the interacton that we obtained with the crystallographic structure for the head and neck squamous cell carcinoma. Each node is an oncoprotein. Each edge is a protein-protein interaction. The color of the node represents the number of pockets that we have detected on the oncoprotein, and the sites represent the number of pockets at the interface with another protein. Focusing on this example, we can see the picadherin dimer. The picadherin is involved in cell adhesion and promotes metastasis and proliferation. These proteins forms a dimer in different steps, and the X dimer, that is one of these steps, has the crystallographic structure available. In the X dimer, we have detected 2 pockets, the orthosteric one in green, that is where the dimerization starts, because a tryptophan on one monomer falls in the hydrophobic pocket of the other monomer. But despite several efforts, no ligand binding these pockets have been found during the last 10 years. Instead, we have found the allosteric site, that is where the dimerization is stabilized by hydrogen bonding. And here there are some ligands with that binds this pocket, inhibiting the dimerization of the picadherin, so having an anti-cancer effect. Second example of allosteric pockets in this case is the colon adenocarcinoma. In this case, we can see the beta-catenin, that is the accumulation of this protein is a K-oncogenic event in the colon carcinoma. And the beta-catenin is interacting with beta-catenin interacting protein and with human T-cell transcription factor 4. The yellow pocket, that is the protein-protein interaction site, is where both partner interacts. But again, in these pockets, despite several efforts, no ligands have been found to bind, to have an effect. Instead, in the small allosteric site in the beta-catenin, there are some binders that have the effect of inhibiting the beta-catenin. And also these ligands, it's very interesting, they are under study for PROTAC optimization. So the final aim is to bring the beta-catenin to degradation. And this example represents our rationale in this work, attempting target triage, but also providing to the scientific community for this complete pocketome in order to help further research in this field. And of course, I would like to thank the Molecular Discovery and Molecular Horizon team and all the collaborators, Ijen and Sonia, and University of Perugia, University of Siena, and University of Bari. And of course, I would like to thank you all for your attention.
Thank you. I think we have time for one question.
Thank you. I was wondering, in one of the first slides, you mentioned that bio-GPS was also used to find hidden pockets, or pockets, I guess. Could you share with how that works? Does it keep track of pocket dynamics? How
Do you mean here?
yeah, a few slides earlier you mentioned things to find hidden sites, but yeah, we just
How bio-GPS detect pockets in general, you mean?
like hidden sites. I guess you pockets.
Sorry?
You
No,
mentioned
here.
hidden sites. I understood that as being like cryptic pockets that don't appear to be there on the crystal structures, but show up in pocket dynamics. But it wasn't really clear to me how this bio-GPS track pocket dynamics.
No, I mean, I mentioned molecular dynamic here because I was saying that when you have a molecular dynamic trajectory, you want to select a representative pocket in molecular dynamic trajectory instead of analyzing the RMSD of the entire protein. That is what you usually do. We can map pockets on the trajectory and then select pockets that are representative of the trajectory instead of the entire, using the entire trajectory.
Okay, and using that, you could find a pocket that shows up in one of the snapshots.
Yeah, instead, yeah, yes.
Right. Thanks, Lydia, again.